everyone, welcome to Ibdp Biology. This is the second part of our first unit Intro to Cell, and today our main focus is about magnification, microscopy, calculation of the magnification. So sit closer and let's get started. Without the invention of microscopy, we would not be able to see or know about the microorganisms and little living things, but just because we have the microscopes, we're able to visualize the structures and properties of the living organisms, so even which is invisible to the naked eye. But we have light microscopes and electron microscopes. Human eye is actually can see something that is visible, but we cannot see something as a unicellular, and we need a light microscope and electron microscope to see and to dive into the cells or the living organisms. Light microscopes are limited in resolution, but electron microscopes have a much shorter wavelengths and they have much higher resolution. What is a resolution, guys? Resolution is actually seeing what is clearly the picture. So with a high resolution, we see the picture in a nice way, but with the low resolution, we see it blurry. But the magnification is defined as magnifying or enlarged image. You have to remember one kind that microscopes are needed to study the cell with a high magnification and resolution are needed to observe cells and their components. Remember this one. First comes the atoms, then molecules, which is equal to 1 nanometer size, membranes 10 nanometer, viruses 100 nanometer, bacteria is 1 micrometer, organelles are up to 10 micrometer, and cells are up to 100 micrometer. You have to remember these things. Remember that bacteria are bigger than the membranes. Viruses, organelles are bigger than the bacteria, and organelles are bigger than the viruses and membranes. Here, calculating magnification. Well, looking at the images, magnification is often stated indicating the scale of the image. So you see the picture, and magnification could be directly stated like multiply 250 times, or indicated by means of a scale bar, which you see on the right bottom of the picture, such as 50 micrometer. This is actually the scale bar. How we are going to calculate the magnification? We will take the magnified size and divide it into actual size. Remember, all units have to be the same if you are using these numbers. So we have got the conversion factors, like 1 meter is 1 meter, 1 centimeter is 10 millimeter, and just those conversions that you can very well understand here. What we're going to do here, we have got the scale bar. We take our ruler. In this case, in these problems, we will always need a ruler. You take your ruler and you put it on this scale bar, you measure it, and we got that it's actually so how much is that? It's 2 centimeter, and it directly converts to millimeter, like 20 millimeter. Then we convert it to a micrometer. It will be 20,000 micrometer. And then what we're doing, 10 micrometer is actual size of the cell, but 20,000 micrometer is actually magnified size. We take the magnified size and divide it to an actual size, and we get the magnification of 2,000 times. So this picture was magnified by 2,000 times here, and we have got some scale bars, and we need a ruler to just put it over that, measure, and find the magnification of certain scale bars here. But what if we do not have a scale bar? How we're going to calculate the actual size? So for this reason, we will directly put our ruler on a picture and measure it here. For example, so if I want to find the actual size, what I'm going to do, I will take the measured length and divide it to magnification, such as here it's written like the picture is about the size of 8 centimeter, which is 80 millimeter, and we see the magnification is up to 90,000 times. So we take the magnification, divide it, and we get the 890 nanometer. But remember that you have to all the time convert the units, because without conversion you will not be able to have a right answer here. Look at this, how we're gonna just measure the pictures without any scale bar here, as you see. Okay, practice. What is the magnification of this image? How I'm going to do this? So we will take the scale bar, we will take our ruler, measure the scale bar, and what we're gonna do, we're going to just take the magnified size and divide it to the scale bar actual size, 5 micrometer, and we will get the magnification here. So here how we're doing this one, guys. Actual size is equal to size in the image divide magnification, or if you want to find the magnification, you will take the size in the image and divide it to an actual size here. How long is the cell shown in the image below here? You can find it here as well. Now we've got some problems to do here. If a red blood cell has a diameter of 8 millimeter and the student shows it with a diameter of 40 millimeter in a drawing, what is the magnification? How are we going to find the magnification? We will take the drawing size, which was 40 millimeter, and just divide it to an actual size to 8 millimeter, and we will type that it was magnified by 5 times. Here you've got some homework problems. A botanist measures a leaf and finds that it is 24 centimeter long and 8 centimeter wide. His drawing of the leaf is 4 centimeter wide, which was the magnification of the length of his drawing, assuming the proportions of the drawing were correct. So you're going to find the magnification. You will take the drawing number and divide it to an actual number here, and you will find the scale and the length here. So we've got some problems here as well. You've got overall 6 problems to solve about the calculation of the magnification or actual size here. Okay guys, thanks for watching. Have a nice day. Bye bye.